Hello students. So in the previous lecture we discussed about the beta oxidation which is carried out in the mitochondria, and the outcome of the beta oxidation is acetyl-CoA, which directly goes into the TCA cycle for the production of ATP. But other than mitochondria, this beta oxidation can also be carried out in another organelle which is called as peroxisome. So but in peroxisome, whatever beta oxidation is carried out, the acetyl CoA which is generated cannot go into TCA cycle, because then there is no TCA cycle in the peroxisomes, and also there is no electron transport chain taking place in the peroxisome. So the so it, so what is the reason why beta oxidation is carried out in peroxisomes? So this is what we are going to discuss today, and it was discovered that all the four enzymes of beta oxidation, they are present in the peroxisomes as well, other than mitochondria. So they have both the mitochondrial as well as peroxisomal like localization. So so how it was discovered that beta oxidation can also be carried out in peroxisome? So it was discovered, the discovery was made when two different types of diseases were studied. One such disease was Zellweger syndrome. So in these, this is again a congenital disease. This is in children who are born with this defect of Zellweger, of not having peroxisomes in their cells. So these children, they don't have peroxisome. And there is another disease which is called as XALD. So this is called as X-linked adrenoleukodystrophy disease. So again, in this is a disease where a specific transporter called as ALD transporter, that is absent on the peroxisome. But the outcome of both the diseases was accumulation of long chain fatty acid. Like, long chain means ki they are beyond 20 carbon fatty acid chain, so they tend to accumulate in the cytoplasm of the cells. And a result of this accumulation, so these children who have this accumulation of these fatty acids, they suffer from several kinds of abnormalities like loss of vision. They have got some behavioral disturbance as well, and most of these children, they die before the age of 10. Although these two syndromes or these two diseases, the origin is different, but they have the common symptoms, and they have the common there is a common marker, which is the accumulation of long chain fatty acid within the cytoplasm. So what this long chain fatty acids does, if they accumulate in the cytoplasm, these fatty acids, they actually destroy the myelin sheath, which is the outer covering of the nerve cell. So as a result of myelin damage, the nerve cells are more prone to damage. So there is the neuronal development of these children does not take place in the way it should happen, right? So as a result of this problem, most of these children, they die before the age of 10. So let's see how the beta oxidation can be carried out in the peroxisome. So like previously we discussed, these free fatty acids, they can travel and they can enter into the cell through specialized fat or CD36 transporters. Once these free fatty acids, they enter into the cell cytoplasm, they undergo activation by acyl-CoA synthetase, so they form a coenzyme A derivative. And in mitochondria, they enter into mitochondria through carnitine shuttle, but in peroxisome, this carnitine shuttle is not required. So there is a special transporter on the peroxisome which is called as ABC transporter, which is full form is ATP binding cassette transporters, or which is also called as ALD protein. So this transporter allows entry of this free fatty acid as a coenzyme A derivative into the peroxisome, right? So the transporter through which these fatty acid enter is also called as ALD transporter, because this is the transporter, or this was the protein which is deficient in XALD patient. So they don't have the transporter, as a result of which these free fatty acids will tend to accumulate in the cytoplasm and will cause damage to the nerve cells, right? And if somebody is not having peroxisome altogether, again there will be accumulation of long chain fatty acid. So why these fatty acids they accumulate in the cytoplasm? The main reason behind that is these very long chain fatty acids, they cannot enter into mitochondria through carnitine shuttle. So they are never oxidized. So they tend to accumulate in the cytoplasm. If there is no peroxisome or if there is no functional peroxisome, this accumulation of free fatty acids, long chain free fatty acid, will cause damage to the nerve cells. Okay, so once these free fatty acid as their coenzyme derivative enters into the peroxisome, it is oxidized by same four reactions that we have studied in the mitochondria. But there are certain differences, like in the first reaction where there is oxidation step involved, where the free fatty acid is converted into trans delta 2 acyl CoA, so metabolite. So as we know that the first enzyme, since it's an oxidation reaction, so these free fatty acid will lose electrons, and where these electrons will go, they will go to FAD, and this FAD will be converted into FADH2. In mitochondria, this FADH2 is converted back into FAD by transfer of electrons to the electron transport chain. But in peroxisome, there is no electron transport chain taking place. So there has to be a mechanism by which this FADH2 is again converted back into FAD, and this is facilitated by a special mechanism which is called as production of hydrogen peroxide. So the name peroxisome actually originates from this prevalence of this hydrogen peroxide within these organelles, and because of this hydrogen peroxide synthesis in peroxisome, it is called as peroxisomes, right? So what happens actually is, as soon as this FADH2 is synthesized, this immediately donates electron to oxygen, and in presence of water, hydrogen peroxide is synthesized. And this is a toxic molecule, and because it can generate reactive oxygen species. So this hydrogen peroxide is quickly neutralized into oxygen and water by a special enzyme which is exclusively localized within the peroxisome, and this enzyme is called as catalase. So catalase will break down this H2O2 into oxygen and water, and that's how the cell will get rid of this hydrogen peroxide, and won't cause any damage to the organelle. So once this trans delta 2 acyl CoA is synthesized, now all other three reaction, which is another hydrogenation, another oxidation, and then thiolase step will take place, and your free fatty acid will shorten by 2 acetyl CoA. So what happens in peroxisome is we just shorten the fatty acid. So long chain fatty acid, they are shortened to around 16 carbon chain free fatty acid, and these 16 carbon free fatty acid then 16 to 14 free carbon free fatty acid chain, they come out of peroxisome, and then now they are free to travel into the mitochondria through the carnitine shuttle for undergoing the process of beta oxidation. So only shortening of fatty acid chain is carried out in the peroxisome. So there is no so whatever acetyl CoA is generated, that won't be utilized for the production of energy. Oxidative phosphorylation में use नहीं होगा उसका, right? So this is all about like beta oxidation in peroxisome. But we also know that there is also a third step of beta oxidation. There will be conversion of one NAD will be converted into NADH. So like in mitochondria, this NADH will go and give its electron to the complex 1 of electron transport chain, but here in peroxisome there is no electron transport chain. So this NADH, it will come out of peroxisome into the cytoplasm, where there are like malate, aspartate shuttle and all. So they will again oxidize this NADH back into they will again like reduce this NADH back into NAD, and this NAD will now travel into the peroxisomes to participate in beta oxidation again. So this is all about like beta oxidation in the peroxisome. But beta oxidation can also be carried out like in plants also. But in plants, the majority of the beta oxidation is carried out in peroxisomes or glyoxysomes. It is not very prevalent in the mitochondria. So the outcome of like beta oxidation in peroxisome is acetyl CoA, but that acetyl CoA is not utilized for the production of ATP, but it is used as a biosynthetic precursors. Okay, so in like peroxisome were actually discovered in the leaf only. So the leaf cells होते हैं उसमें जो peroxisomes होते हैं, जो organelles होते हैं उन्हें peroxisomes बोलते हैं. Like these peroxisomes are present in the germinating seed, then they are called as glyoxysomes. So whatever acetyl CoA which is involved in the glyoxylate cycle is generally comes from the beta oxidation of the um, of the fatty acids which are stored in the seeds itself, right? So 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 there in plants, whatever acetyl CoA is produced, it is used for the generation of like metabolites like oxalate or malate, or it can also be used for gluconeogenesis or other processes where it can be utilized for the production of glucose and sucrose, which are used as storehouse of energy within the plant cells. So this is all about like peroxisomal beta oxidation. So I would like to stop here. Thank you.